So let's talk about organic compounds as opposed to inorganic compounds. Organic compounds are going to be molecules that are unique to living systems that contain carbon. We could say, with exceptions like carbon dioxide, CO2, or carbon monoxide, CO, those are inorganic. For that reason, I like to lengthen the definition of organic compounds into molecules unique to living systems that contain carbon in combination with hydrogen. That immediately would let us know that carbon dioxide and carbon monoxide are inorganic. Examples of organic compounds, also known as biological molecules, are carbohydrates, lipids, proteins, nucleic acids. Many of these are polymers made up of repeating or similar units. Actually, all of them but lipids are made up of monomers or building blocks that repeat to build polymers. The polymers are made by dehydration synthesis and then broken down by hydrolysis reactions. Let's look at carbohydrates first. They include sugars and starches and contain carbon, hydrogen in combination with oxygen. And hydrogen and oxygen are going to be in a 2-to-1 ratio in carbohydrates. There's three classes of carbohydrates: single sugars, two sugars, and many sugars, or monosaccharides, disaccharides, and polysaccharides respectively. The monomer of a carbohydrate is a monosaccharide, which is a single simple sugar. Examples include glucose, which is very common, and then fructose, galactose, deoxyribose, and ribose. If you put two monosaccharides together, you get two sugars bonded together, and that's a disaccharide. If you have many sugars bonded together, so more than two, you're looking at a small polysaccharide to very large molecules like starches. Now, double sugars are when we again combine two monosaccharides, like glucose and fructose to give you sucrose, galactose and glucose to give you lactose, and glucose and glucose or two glucose monomers to give you maltose. Much bigger though when we're looking at polysaccharides, so polymers of simple sugars, polymers of monosaccharides. Starch is actually a carbohydrate storage form that's used by plants, whereas glycogen is a carbohydrate storage form used by animals. Our glycogen is actually stored mostly in the skeletal muscle and liver cells. And these big molecules, these biological organic molecules, they're not going to be very soluble. Here's an example of a glycogen with many units of sugar as glucose that are actually connected here by glycosidic bonds, holding them together. But they could be broken apart to provide readily usable and quick energy to the blood so it could get to cells. Next, we can look at lipids, which are also insoluble in water. Lipids probably even more so than carbohydrates. Just like carbohydrates, they contain carbon, hydrogen, and oxygen, but in different proportions. The main types of lipids are triglycerides, phospholipids, and steroids. We're going to talk about triglycerides first. They're composed of 3 fatty acids and 1 glycerol molecule. The fatty acids are made up of carbon and hydrogen, so they're called hydrocarbons. And the 3 fatty acids is where we get the tri part of triglyceride. The glyceride part comes from glycerol, which is a sugar alcohol. And we have some similar functions of lipids as we see with carbohydrates. They're both used for energy production. But lipids are actually long-term energy storage and have to be broken down through a longer series of steps to actually provide usable energy, whereas carbohydrates are ready and willing to provide quick, usable energy. Lipids can also provide insulation and protection in the human body. When we're talking about fatty acids, when we're talking about triglycerides, we're actually considering different types of fatty acids. There's saturated fatty acids and unsaturated fatty acids. With saturated fatty acids, they have single bonds and they're solid at room temperature. So that's a lot of S's. Saturated, single, solid. And that's one way to keep the information in your brain. The single bonds between the carbon atoms that make up the hydrocarbons that we find in fatty acids have the maximum number of hydrogen bonds connected. That allows these triglyceride molecules with their hydrocarbon chains to fit very closely together if they're saturated fatty acids. And when molecules fit closer together, we're more likely to see a solid. With unsaturated fatty acids, we have one or more double bonds between the carbons, so we have a reduced number of hydrogen atoms. That also produces a kink or a bend in the fatty acid tails or hydrocarbons that make up these triglycerides. That's not going to allow these particular tails of a triglyceride to fit very closely together, and when atoms don't fit as closely together, they're more likely to be a liquid. So unsaturated fatty acids have double bonds at least one or more, and they tend to be liquid at room temperature, like plant oils, like olive oil. So here's a basic formation for a triglycerol molecule. On the left, we see glycerol and 3 fatty acid chains. Those fatty acid chains are made up primarily of carbon and hydrogen, and you can see that the removal of H2O, or one hydrogen atom and one hydroxide atom, so the removal of water 3 times, is going to be utilized to put together, or dehydration synthesis reaction is going to occur here, and put together the glycerol molecule and the 3 fatty acid chains. That's going to give us a triglyceride, or neutral fat. When it comes to phospholipids, we can actually see for the first time a saturated fat and an unsaturated fat. Look at the image on the top right. The straight fatty acid is saturated, and the bent one is unsaturated. So you can see that the arrangement would prevent packing if they were all unsaturated. For a phospholipid, we see those 2 different types of fatty acid tails. Still the glycerol backbone, but in addition we haven't seen yet, which is a phosphorus-containing head group. This head group is going to be hydrophilic. It likes to hang out near water, whereas the component that has glycerol and fatty acids is hydrophobic, or essentially scared of water. That's going to be important in cell membrane structure because the arrangement of phospholipids creates the cell membrane. Generally, what we see in a cell membrane is 2 layers of phospholipids, with their head regions facing either the outside or inside of the cell, and their tail region on the inside sequestered away from watery environments. And last but not least, for another type of lipid is steroids. Steroids consist of 4 interlocking ring structures, and common steroids are like cholesterol. Cholesterol is going to be kind of the basic viewpoint for how to build different types of hormones, like the steroid hormones testosterone and estrogen. They would just have slightly different attached groups. But by far the most important steroid is cholesterol because it's a building block for vitamin D. It's going to help with steroid synthesis. It helps with bile synthesis, and we're also going to find it as an important component of the cell membrane. And next we're going to be looking at proteins. So we're going to take a break here between carbohydrates and lipids, which are great for energy storage or production. And then we're going to move on to proteins, which are made up of amino acids, and then later look at nucleic acids.